What is hemoglobin? We have already read in physiology. Hemoglobin is a protein and it consists of a heme part and a globin part. This globin part is the protein chain and this heme part is the iron part. So, hemoglobin actually constitutes approximately 90% of the dry weight of a mature RBC. RBC is nucleated or non-nucleated? Non-nucleated. Mature RBC is non-nucleated. So, the volume of the RBC is 90% of the dry weight of the RBC is hemoglobin. So, it consists of heme and globin. So, this is the structure. The hemoglobin molecule is a tetramer, means it has 1, 2, 3, 4 subunits. Two pairs of similar polypeptide chains called globin chains. So, there are two pairs. So, this is an adult hemoglobin structure. These two pairs, these are beta chains and these are alpha chains. Okay. So, this is the structure of adult hemoglobin, two beta chains and two alpha chains. So, there are four chains and in which the heme iron is attached in the form of ferrous form. Iron is present in the ferrous form, not in the ferric form. Ferrous form and protoporphyrin. So, these are the iron that is attached to the globin chains. So, there are four sites and all these are ferrous iron 1, 2, 3, 4. This 4 heme group is attached and there is the attachment of the iron. And 96% of the adult hemoglobin is HbA. HbA means the adult hemoglobin and this adult hemoglobin has 2 alpha chains and 2 beta chains. This is represented as alpha 2 beta 2. This is the normal structure of adult hemoglobin. So, there are many types of hemoglobin, some of them physiological, does not affect anything, some of them are pathological. So, you will come to read about the diseases in your theory classes. So, the hemoglobin variants. The adult type of hemoglobin is alpha 2 beta 2, this is present in adult. Then fetal hemoglobin is alpha 2 gamma 2, this is present in the fetal life. Not in the embryonic life. In the embryonic life there are various hemoglobin present like hemoglobin Portland, Gower 1, Gower 2. Okay. So, these are present in the embryonic life. Okay. You have to know the names only, you do not have to go into the details. Just hemoglobin Portland, hemoglobin Gower 1, Gower 2, these are present in the embryonic life. And in the fetal life, this is fetal hemoglobin alpha 2 gamma 2. This beta chain is replaced by the gamma chain. The alpha chain remains the same and this beta chain is replaced by the gamma chain. That is alpha 2 gamma 2. This is present in the fetal life and for some time after birth and then it completely gets converted to this adult hemoglobin. Okay. At the 8th week of development, these Gower 1, Gower 2, Portland, these are gradually replaced by fetal hemoglobin or HbF. That is alpha 2, not it is wrongly written here, it is alpha 2 gamma 2, not beta 2, alpha 2 gamma 2. And this remains the predominant hemoglobin after the birth and it gradually decreases and at 30 weeks of age, 30 weeks means like 7 months like that, it decreases to less than 2% and the rest of the hemoglobin is HbA, that is alpha 2 beta 2. Hemoglobin, fetal hemoglobin is found up to 30 weeks, then it goes down to 2% by 30 weeks. And after 30 weeks, at 30 weeks, you will get the adult hemoglobin. 98% will be the adult hemoglobin. And there is also another hemoglobin, that is HbA2. A2 is alpha 2 delta 2. This HbA2 is present and it is normally in very less quantity it is present. So, the main hemoglobin in a healthy mature adult is HbA predominantly and some amount of HbA2. The hemoglobin, fetal hemoglobin decreases to less than 2% by 30 weeks, and in adult it is almost negligible. The main hemoglobin is HbA and some amount of hemoglobin A2, less than 4% or like that HbA2. And apart from that, there are some abnormal hemoglobins which causes disease in a person. These are sickle cell anemia, HbS, HbC, HbD, HbE. These all are abnormal because they cause disease manifestation within the person. So, these are the various kinds of hemoglobin. What is the function of hemoglobin? This hemoglobin is present in the RBC and these are the 4 heme moiety and these are the alpha chains and beta chains. So, what happens? Heme has the ability to bind oxygen reversibly and carry it to tissue. It also facilitates the exchange of carbon dioxide between the lungs and the tissues. Thus, hemoglobin functions as the primary medium of exchange of oxygen and carbon dioxide. So, hemoglobin does both the things. In the lungs, it traps oxygen from the inhaled air, it goes to the tissue, there it releases the oxygen to the tissue, and from tissue it brings the carbon dioxide to the lung and it releases the carbon dioxide into the lung. So, hemoglobin does both the functions. That is why hemoglobin is so important. Okay. It carries hemoglobin, it carries oxygen to the tissues and it also facilitates the exchange of carbon dioxide between the lungs and the tissues. It is the primary medium of exchange of oxygen and carbon dioxide. You have read this, no, in physiology? So, normal hemoglobin concentration in humans, it is given in gram per liter, 150 plus minus 20 gram per liter. If you convert it to DL, it will be 15 plus minus 2 gram per DL. Okay. In case of men, this is the WHO standard. Men is 150 plus minus 20 gram per liter or 15 plus minus 2 gram per DL. When you convert liter to DL, automatically you have to shift 1 0 towards the left. So, it will be 15 plus minus 2 gram per DL in case of men, and in case of non-pregnant healthy woman it is 13.5 plus minus 1.5 gram per DL or 135 plus minus 15 gram per liter. So, this is the normal value. So, if someone asks you the normal value of hemoglobin, you have to mention it in this way. The normal value in men is this, the normal value is in non-pregnant woman is this. In pregnant woman, the value differs. In the first trimester, it is like 12.4 plus minus 1.35 gram per DL. In the second semester, it is 11 plus minus 1.17 gram per DL, and in the third trimester it is like 10.6 plus minus 1.09 gram per DL, like this. In pregnancy, the hemoglobin level drops because pregnancy is a negative iron balance situation. So, there will be gradual drop of hemoglobin, and in babies the hemoglobin level is very high. You know, when a baby is born, its hemoglobin level is like 18 plus minus 4 gram per dl. A baby has this much of hemoglobin. By the third day, this hemoglobin, these RBCs breaks down and the hemoglobin part is released and the baby gets jaundice, which is known as physiological jaundice. And then this 18 gram per dl, that will gradually reduce. So, at birth it is at its peak 18 plus minus 4 gram per dl. So, this is this dense. What are the physiological and condition when there is variation in hemoglobin concentration? We read about the normal values. Now, these are the physiological conditions where there is variation in the hemoglobin concentration. In elderly patient, there will be the hemoglobin will drop. In renal insufficiency, inflammation, testosterone deficiency, diminished erythropoiesis, myelodysplasia, all these things are responsible for the decreasing hemoglobin level in case of elderly. Exercise. The more you exercise, it will increase the hemoglobin concentration. Posture. Lying to sitting increases hemoglobin concentration. If you tell the patient to lie down and then you collect the blood for hemoglobin, it will be something less than if you take the blood in sitting position. We always prefer to take blood in a sitting position unless the patient is bedridden. Now, altitude, you all have learnt about altitude sickness. People who live in the higher altitude areas, they have increased hemoglobin concentration so that they can carry more oxygen. In the higher altitude, the oxygen concentration in the air is low. So, the hemoglobin has to carry more oxygen. So, there is increased concentration of hemoglobin. Smoking also increases the hemoglobin concentration, but it is not advocated to increase hemoglobin concentration by smoking. No one will say that you have, oh, your hemoglobin level is low, so go and smoke and increase your hemoglobin level. This is not done, but smoking does increase the hemoglobin level. Smoke, your GFR will increase and your hemoglobin concentration will increase. Will anyone tell you? So, smoking causes increase in hemoglobin concentration, but this is not advocated. Nobody will say, oh, you have low hemoglobin and go and smoke. This is not done that way. So, what are the conditions when a hemoglobin estimation is indicated? So, you have to determine the presence of anemia. This will come to you as OSPE. A person, suppose 30-year-old female comes to you with fatigue, headache, dizziness, and she comes to you and says that I am not feeling well, I have fatigue, dizziness, I have frequent headaches. So, when you do the general checkup, you see pallor, the patient is pale, you see the under the eyes, you see the lower part of the conjunctiva and you see it's pale white. So, from the clinical examination, you can say that the patient has pallor. When you can see clinically that the patient has pallor, obviously you have to determine the severity of anemia and you have to go for an hemoglobin estimation. Screening for polycythemia. Polycythemia is also like in babies the hemoglobin is 18 gram per dl like that. In polycythemia also the hemoglobin in case of adult is like that 18 gram per dl like that. So, this is called polycythemia. So, if you are screening for polycythemia, you have to see the hemoglobin and PCV. There also you have to do a hemoglobin estimation along with a PCV estimation. And response to specific therapy in anemia. A patient is already anemic, you have diagnosed that the patient is anemic. Now you have put her on iron tablet, iron folic acid tablet, or you have put her on vitamin B12 folic acid tablet. So you have to see the response to therapy. Otherwise, it is no use giving the therapy. You have to monitor that the patient's hemoglobin level is increasing or it is static. You have to see that. So, to see the response to the specific therapy of anemia, you have to do the hemoglobin estimation. Estimation of red cell indices. What are the red cell indices? Can you tell me? Red cell distribution weight. So, red cell distribution weight does not come under the red cell indices, and you can't determine the red cell distribution weight manually. You have to take the machine thing. The machine will tell you the red cell distribution weight is this, and it is normally 12 to 16. So, red cell distribution weight basically tells you the amount of anisocytosis and poikilocytosis. If see, as per the hemoglobin concentration of the cell and the size of the cell, there may be microcytic hypochromic, then normocytic normochromic, and macrocytic cells. So, the proportion of these cells, they may be equal, then you will get a normal RDW, and they may be unequal. There may be mixed population of cells, some are very microcytic, some are normocytic, some are macrocytic, then you call it a anisocytosis, difference in the size of the cells, and poikilocytosis is difference in the shape of the cells. RBC is a biconcave disc, but in the pathological conditions what happens? You will see pencil shaped cells, you will see bite cells, you will see cystocytes, you will see macrovalocytes. So, all these things contribute to the red cell weight. RDW, high RDW means there is more amount of anisopoikilocytosis. So, the red cell indices, this includes MCV, MCH, MCHC, and PCV and hemoglobin are required to measure these red cell indices. That is why you have to do the hemoglobin estimation, the PCV estimation, then only you can find out the red cell indices. And in the blood bank, obviously for the selection of blood donors, you have to first of all you have to see the blood donor has optimum level of hemoglobin or not. If the blood donor himself or herself is anemic, then how can you take out a blood? So, 12.5 is the cutoff in case of blood donation. 12.5 gram per dl is considered the cutoff. If the hemoglobin is below that, you don't take the blood for donation. If it is above 12.5, you take the blood for donation. Okay, so for selection of blood donors, you also have to do the hemoglobin estimation. Now, what are the methods? The methods may be colorimetric methods, gasometric methods, and chemical methods. Okay, and also specific gravity method, this I will come later. So, colorimetric methods, this is the most common thing, most common method which is used. The machine uses a different technique. So, there are some visual methods. So, these visual methods are Sahli's acid Hematin method, then Talquist hemoglobin chart, WHO hemoglobin color scale, oxyhemoglobin method, specific gravity method. These are just visual methods. Here you have that Sahli's hemoglobinometer. Sahli's hemoglobinometer is actually this acid Hematin method. Okay. So here this is the box, Sahli's hemoglobinometer. So the colors are given here and comparator box. Here you put this like this and you have to compare the color of this with the color from this side. Okay, you have to compare, and by seeing it with your naked eyes you have to compare the color and then you can give the hemoglobin percent from here. Here is the reading. Okay, this you will have to do by yourself. So the next class you will do it by yourself. This is Sahli's acid Hematin method. This comparator box and these things are used for Sahli's acid Hematin method. Then there is a Talquist chart, hemoglobin, WHO color scale, oxyhemoglobin specific gravity method. There are photoelectric methods, cyanohemoglobin method, oxyhemoglobin method, alkaline Hematin method. Gasometric method, this is totally obsolete, this is no longer used. This is very time consuming and expensive, and the result is there is a variant of 2%. There is 2% variation. So this method is almost obsolete. You can take the screenshot if you want, if it is asked that other method, other method, you tell all the methods, and when no method is left then you can tell this gasometric method. This is not used for practical purposes. And chemical method, this is also a time consuming method, this is also not used in this way. First you have to measure the iron content, then you have to count, then you have to calculate and all these things. This is also not a feasible method. You can do a hemoglobin estimation if you have the cell counters in your lab. You can do a hemoglobin estimation in 5 minutes, they will give you the result. So these are all of historical value. Specific gravity method, it roughly estimates from the specific gravity of the blood. This method is useful, this copper sulfate technique. This is a method which we use in the blood camps. In the blood camp there are 80, 90, 100 donors waiting to give their blood. So it is not possible to check everyone's hemoglobin. So what do? You prick and put the drop of blood on the copper sulfate solution. If it floats on the copper sulfate solution, it is less than 12.5 gram, and if it sinks, it is more than 12.5 gram. So this copper sulfate technique, this is used in mass screening like in a blood donation camp. Okay. Otherwise it is not used for routine purposes in the lab. Its importance is only in blood donation camps. In copper sulfate method, you have to remember this thing. The solution of the copper sulfate will have a specific gravity of 1.053. This thing you have to remember. This is asked, what is the specific gravity of the copper sulfate solution? Okay. And this specific gravity is equivalent to 12.5 grams. Okay. So if the blood drop sinks in this 1.053 specific gravity of copper sulfate solution, then it means that it is more than 12.5 gram per dl. This figure you have to remember, this figure you have to remember. Okay, this is the copper sulfate method. Now also when you go to the far villages in the blood camps, still this method is used. So this method is to some extent relevant. This is the specific gravity method. This is the picture. Then this Sahli's acid Hematin method. Here we use blood is mixed with acid solution so that hemoglobin is converted to brown colored acid Hematin. Acid Hematin method. Acid solution, mostly HCl, and blood is converted to brown colored acid Hematin and it is diluted with water till the brown color matches that of the brown glass standard. HB value is read directly from the scale. So here you have seen you put this here, that acid Hematin, after the acid Hematin is formed, you mix blood with this acid and then you add water by dropper. You add water drop by drop until you match these colors. Okay. On the next class you will do it by yourself. Then what do you need? Sahli's hemoglobinometer, Sahli's pipette. It is marked at 20 microliter or 0.02 ml. There is the marking. You have to take the blood up to this 0.02 ml marking. Then there is a stirrer, there is a dropping pipette and the reagent is N/10 hydrochloric acid and distilled water. Okay. So this is the stirrer, this is a glass rod, this is the hemoglobin pipette. From here you have this, there is the mark. Okay, 0.02 ml. You draw blood up to this and put it in this Sahli's graduated hemoglobin tube and you add N/10 hydrochloric acid. N/10 hydrochloric acid you add to this, you mix it properly and then there is the standard, you put this graduated tube in this and you add distilled water gradually and when this color matches with this, then you directly read from this graduated tube, you directly take reading from this. This is the whole method, N/10 HCl into hemoglobin tube up to 2 grams. Blood sample in Sahli's hemoglobin pipette up to 20 microliter or 0.02 ml. Then the blood sample is added to the acid solution. First the acid solution is taken, then the blood sample is mixed with the acid solution. Don't say that we take the blood first and then we put the acid. No, we take the acid solution first, then we put the blood in the acid solution. Then it is mixed with the stirrer and it is allowed to stand for 10 minutes for the reaction to happen. And then distilled water is added drop by drop till the color of the solution matches the brown glass standard. The reading of the lower meniscus of the graduated tube is taken in grams. See, the fluid never stays like this, the fluid stays in a concave way. In any tube you put plain water also, the level is never like this, it is like this, concave. So you take the reading from the concavity. Where there is the concavity, you take this lower point as your reference and you take the reading from there, and it will be from the graduated tube and the reading will come in grams. So, what are the advantages? It is easy, quick, inexpensive, can be used as a bedside procedure, and it does not require technical expertise. These are the only advantages. You can, a patient has come to you, patient is very sick, if you send you have to draw the blood and send the blood to the laboratory, wait for the results, and you have a strong clinical suspicion that the patient is anemic, you have to do a blood requisition. So, you can do this in the bedside and it is very quick, it is very easy to perform, anyone can do that, and on the basis of this you can go for a blood requisition. And the report that comes from the laboratory, it will take at least 20 to 30 minutes it will take. First you have to send it to the lab, and then from the lab you have to get the report. So, it is a long process, relatively long process, but it is very quick, you can do it in the bedside. That is why this method is still in the syllabus, otherwise it would have been kicked out of the syllabus. Now, the disadvantages: for maximum color, longer time is required. Perfect matching with brown glass standard is not possible. See, it is you do it by chocometry, just see and compare. So, there will be human errors. One of you will see that the color is not matching, when I need to drop 2 or 3 more drops of distilled water. Another one will think, no, the color is exactly matching, there is no need to dilute it anymore. So, there will be person to person variation. In any case where you are taking the reading by your bare eyes, there is this problem. It will vary from person to person, there is no standard, and the same thing when you repeat again, it is not reproducible. After 5 or 10 minutes, if you do from the same patient, if you take the sample and perform the same method, you might see that the difference in the reading happens. So, this is every time you give the blood and you will get the exact result, it is not possible. This is the main drawback. And carboxy hemoglobin, methemoglobin and sulfhemoglobin are not converted to acid hematin. Okay. So, these hemoglobin abnormalities, if are there, they will not be converted to acid hematin. So, you do not know what the patient is having. So, if the patient is having any methemoglobinemia, if the patient is having methemoglobinemia, then it will not be converted to acid hematin, then you can't measure the hemoglobin level of the patient. Development of the color is slow and acid hematin is not stable. You have to take the reading and you have to keep the reading, and if you leave the acid hematin, it will degrade. And if someone just does the test and leaves it in the comparator box and goes away and comes after giving round for 30 patients and comes to see what is the reading, it will definitely degrade and you will not get the exact level of hemoglobin. It is not a stable compound, and the source of light will influence the comparison of the colors. If you take it in the sunlight, it will give one type of reading. If you take it in the room in the artificial light, it will give another kind of reading. So, these are the disadvantages of acid hematin method, and due to all these things, this 5-part 3-part all this came into action. Cyanmethemoglobin method, and cyanmethemoglobin method, this is the method which is followed everywhere. even within the hemocytometer, the counters, actually this method is used. It is the most accurate method and it is recommended by International Committee for Standardization in Hematology because all forms of hemoglobin are converted to cyanmethemoglobin except sulfhemoglobin, and it is stable and reliable standard is available. So, what is the main advantage of cyanmethemoglobin method? It is very accurate. It is very accurate and all the forms of hemoglobin will be converted to cyanmethemoglobin, and sulfhemoglobin, this is very rare. Leaving this condition, all of the hemoglobin that is there in the body will get converted to cyanmethemoglobin, and this is a stable compound. Cyanmethemoglobin is a stable compound and you can make reliable standard from this. And what you do, it is this method is used in the lab nowadays also. This is very much in use in the labs. Small labs which cannot afford to buy a cell counter, they use this method for estimation of hemoglobin. What do you do is, there is a solution called Drabkin solution. Blood is mixed with Drabkin solution. What is Drabkin solution? You have to memorize this. What is there in the Drabkin solution and what is its pH? Its pH is 7 to 7.4. It has potassium ferricyanide, potassium cyanide, potassium dihydrogen phosphate, non-ionic detergent and distilled water. These are the compositions of Drabkin solution. Any question? So, what happens? When you mix blood with this Drabkin solution, the erythrocytes are lysed, producing an evenly distributed hemoglobin solution. Potassium ferricyanide converts hemoglobin to methemoglobin. Methemoglobin combines with potassium cyanide to form cyanmethemoglobin. Okay. The first thing is potassium ferricyanide. So, first of all, when you mix the Drabkin solution with the blood, then what happens? There is distilled water, it causes lysis of the RBCs, and when the RBC gets lysed, the hemoglobin comes into the solution. Then potassium ferricyanide acts with hemoglobin and converts it into methemoglobin, and this methemoglobin combines with potassium cyanide to form a cyanmethemoglobin. All hemoglobins present in the blood are converted to this form. Absorbance is measured in a spectrophotometer at 430 nanometer. 540 nanometer. The colorimeter that we use, that has a spectrophotometer of 540 nanometer. 540 nanometer. To obtain amount of unknown hemoglobin sample, its absorbance is compared with the standard cyanmethemoglobin solution. So, first of all, you prepare the standard. There is a standard, and you take the hemoglobin of a person whose hemoglobin you do not know. You process the whole thing like this, and at 450 nanometer, you compare the patient sample and the standard, and from the standard and the patient samples reading, you can determine the patient's hemoglobin. So, this is the most accurate measure for hemoglobin. So, how to calculate the hemoglobin? you have done all this thing is done. And how to calculate the hemoglobin? That is absorbance of the sample by absorbance of the standard into standard concentration in gram per dl. You will get, there will be a reading, absorbance of the sample, suppose it is 21 and absorbance of the standard it is 24, then 21 by 24 into standard concentration, that means you have the standard, you get the standard in the package, into the standard concentration per gram per dl, and the result comes in gram per dl. This is the calculation. You have to remember this calculation. This is the whole procedure, step by step. 20 microliter blood in 5 ml of the Drabkin solution. You mix it well, leave it for 5 to 10 minutes, and it is measured by spectrophotometer at 540 nanometer. Okay, this standard solution is provided by with the kit, and here is how you calculate the solution. This thing will be asked in the examination. You have to show this Sahli's hemoglobinometer, this acid Hematin method, and this is the method that you will be asked in the hemoglobinometer. How do you do a Cyanmethemoglobin method, and how do you calculate the hemoglobin from the Cyanmethemoglobin method? Here you get a calculated value, there you are getting a compared value, you are just comparing with your eyes. Here you have to calculate the value of the hemoglobin. So when you do a calculation, it is always better. And this is the photoelectric meter. Here you get the reading, and here you place the sample. Okay, this will be there in the department also. I have to see. The same thing written twice. You just go through this thing you remember by heart, and these are all elaboration of the same method. And if you use a diluent, you have to multiply it with the dilution factor by 100. Now, when will you get erroneous results in Cyanmethemoglobin method? What are the drawbacks? Hypertriglyceridemia, lipemic serum, white serum. The serum is normally straw colored. If you get white serum, that is lipemic solution, lipemic blood. Leukocytosis, the leukocyte count is very high. Plasma cell dyscrasia, and these conditions will cause erroneous method by Cyanmethemoglobin method. And this method is, this Cyanmethemoglobin thing is very stable, and if you keep it for half an hour and you come and take the reading after half an hour, it will give almost the same reading. It is a stable compound, it will not decay. So these are the advantage. All forms of hemoglobin are converted to cyamethemoglobin except sulfhemoglobin. Visual errors are not there as there is no color matching. It is a stable compound. If you can't take the reading immediately, you can wait, and absorbance may be measured soon after dilution. You don't have to wait for much time, and it is a reliable and stable, and is available from World Health Organization for direct comparison. Now the disadvantages. Diluted blood has to stand for a period of time to ensure complete conversion of hemoglobin. Diluted blood. Okay, sometimes you get diluted blood. I told you on blood drawing that if someone draws from the cannula through which the saline is passing, then this diluted blood will have to stand for a period of time to ensure complete conversion. It contains, the Drabkin contains potassium cyanide. So, the Drabkins is a poisonous solution. And you will never pipette by mouth. Okay. You will never pipette Drabkin by mouth. The rate of conversion is containing carboxyhemoglobin. If there is carboxyhemoglobin, then the reaction takes prolonged time. If there are plasma proteins which cause increased turbidity, then there will be abnormal prolongation. High leukocyte count, there will be abnormal prolongation, and this will also interfere with the reading of the Cyanmethemoglobin method. These are the disadvantages. So, this is the 3-part counter. It has two chambers, one is hemoglobin WBC chamber and another is RBC platelets chamber. Here you will get a graph like this and you will get all the parameters MCV, MCH, MCHC, RDW, hematocrit, platelet, hemoglobin, and the WBC, total WBC count you will get, total platelet count you will get, but the WBC differential count will come like neutrophils, lymphocytes, others. In the 3-part counter it comes as others, and others means monocytes, eosinophils, and maybe if you get nucleated RBCs also they will come under others. And this is a 5-part analyzer. All the parameters are same except you will get neutrophils, eosinophils, basophils, lymphocytes, monocytes. You will get the total differential count and as well as the absolute values. So, this is the difference between the 3-part counter and the 5-part counter. And the 5-part counter is, since it is an advanced machine, its results will be more advanced. This will give you the flags like platelet clumps present, like immature granulocytes present, then NRBC present. You will get all these graphs there, you will get all these flags. In the printout which comes out, you will get all these flags. There is a column for RBC flag, WBC flag, platelet flag. You will get all the anomalies the machine can detect and you will get those flags and you will become cautious and you will see the slide. When you see the slide, you can always compare the slide with the readings given by the machine. This is the 5-part differential. Now, this Talquist hemoglobin chart, this is done where there are no means of doing hemoglobin in any way. In a far away village where there is only a village sub-center, the pregnant mothers are coming, how do you do a hemoglobin assay? There is nothing. You won't even get a proper road to reach to the sub-center. So, a Talquist paper is there, you prick the finger and place the blood drop on a absorbent paper and the color is matched against the chart, and then the reading is taken and obviously it is such a method that there is chance of error, 20 to 50% chance of error is there, and you do it in very remote areas where there is nothing there. You take this, carry these papers with you and you can do it, but there is high chance of error. This is what the Talquist paper looks. It is of no importance in a tertiary care center, but at the sub centers they do like that. They match the colors with this. Okay, and gross level is there. If your blood falls there, there will be a level. Hemoglobin is from attogram to attogram, then if this color matches, then hemoglobin level is from between this gram to this gram, like this. Now WHO has also a color scale. This is also for the far away areas. And it is also used for screening women and children's health in the faraway areas and starting iron therapy. It can also be used in blood donation camps, where we have to do screening of 125 patient donors like that. Then hemoglobin color scale is a useful method. Okay, its efficacy is greater than Talquist method. This is the hemoglobin color scale. This is the scale 14, 12, 10, 8, 6, 4. This is like that. Okay. So, this is the summary of the class. What you have to know is the values, and you have to add the pediatric value also, or the value of newborn also. Sahli's acid Hematin method you have to learn. Cyanmethemoglobin method you have to learn. What is Talquist hemoglobin chart you have to know. What is WHO color scale you have to know. And you have to practice this Sahli's acid Hematin method you have to practice. 5 ml of Drabkin solution and to 20 microliter of blood. There are measuring pipettes. You don't take a dropper and give the drop of blood like. There are my pipettes. You have to adjust the pipette to 20 microliter. Then you draw the blood and place it in the Drabkin solution. Okay. Then the reaction occurs and you place it in the colorimeter. And that colorimeter, you have, first of all you have to standardize the colorimeter. You have, the standard is provided along with the solution. Okay, and then you have to proceed. Okay, and this thing you will do it in the next class. Practice. You have to do this one in your exam, and all what I taught you today, this will be asked in the Viva table. These instruments will also be there in the Viva, and they will pick the comparator box and say what is this, in what method you use it, how do you do the test. This will be there in the instrument table also. Okay.